Hello students, good morning. I'm your teacher Dolcy, and today, as you know, we are on chapter number 2 of unit 3rd, that is Biomolecule, and we have to discuss today the classification of amino acids, means how we can classify amino acids based on various types. As I told you in the last lecture, firstly we are going to classify amino acid based on their chain and polarity, means they are said to be polar or said to be non-polar depending upon the charge on the chain. Okay, and the polar one are said to be the hydrophilic because the polar compounds are soluble in water, and the hydrophobic compounds are said to be non-polar because they are not soluble in water. So hydrophilic says to be hydro means water, philic means loving, so they are water loving molecules, and hydrophobic are water repelling. Phobia means fear, so they are water repelling molecules. Further, the polar amino acids are of three kinds. They can be neutral if they do not carry any charge, means they carry equal positive and negative charge. Then are the acidic amino acid which have an excessive COOH, that is carboxylic group, due to which negative charge is present on the amino acid. Third one are the basic amino acids. They are the amino acid which carry excessive or extra NH2 groups. And as you know, NH2 means amino, ammonio. From that it carries the positive charge. So polar amino acids are of three kinds: neutral one, acidic one, and the basic one. Neutral one are those which do not carry any charge, acidic one carry negative charge, and basic one carry positive charge. Next classification is on the non-polar basis or the hydrophobic amino acids. They can carry an aliphatic chain, that is alkyl chain, or can be aromatic. So they are of two type, aliphatic and aromatic. As I told you in class 10th that aromatic are those which carry the ring of benzene in their chain. They are two, phenylalanine and tryptophan, and other are alkyls. Alkyl are those which carry a long hydrocarbon chain, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, in this way. And the chain is not in the form of ring, that is aromatic. They are said to be the aliphatic or alkylic type of amino acids. Okay. So now it's done to explain the structure of all these amino acids based on the charge. First classification is the polar and neutral amino acids. Polar and neutral amino acids. So they are the amino acid which carry neutral chain, which do not carry any charge. And they are polar means they are soluble in water. So polar neutral amino acids are four: asparagine, glutamine, serine, and threonine. And you have to memorize the chain of all these. In asparagine, CH2, C double bond O, NH2 R group is present. In glutamine, the chain is increased, or its homologous series of the asparagine. It carries CH2, CH2, NH2 and C double bond O. Okay. Next is the serine. As you know, it is alcoholic, so it carries CH2OH group. Next one is the threonine. It is also the homolog of the serine, so it carries CHOH and the CH3 group. Second one are the polar and the charged. As you know, they can be of two type, positively charged or negatively charged. If they are positively charged, they are three: lysine, arginine, histidine. And if they are negatively charged, they are two: aspartate and glutamate. Aspartate is CH2COO negative, okay, and its homolog is the glutamate, which carry excessive CH2 group, so it is CH2CO negative. Next are the positively charged. There are three: lysine, arginine, and histidine. Lysine and arginine are homologs of each other. They represent CH2, 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 and NH3 positive, excessive NH3 positive in the neutral one. Next, the asparagine, it carries one more NH3 group. It carries CH2, CH2, CH2 three times, then NH, then C, double bond NH3 positive, and NH2. In this way, the excessive positive charge is present, hence they are said to be the positively charged amino acids. Next classification, it is based on the sulfur containing amino acids. So, as you know, the amino acids that carry the sulfur group in their side chain are only two. First one is cysteine, second one is methionine. Cysteine is actually a polar amino acid. We can consider it also there, but here I consider it in non-polar just to make you explain that is sulfur containing. Actually, it is polar amino acid. Okay, so cysteine, it carries CH2 SH group, and methionine, it is the homologue, so it carries CH2 CH2 SCH3 group. Okay, so next compound, that is of non-polar amino acids. So non-polar amino acids are of two type, aliphatic and aromatic. Firstly, we have to memorize the structure of aliphatic one. As I told you in the previous lecture, I explained you the diagram. These are glycine, which carries the H as R group. Second is alanine, which carries the CH3 group as the R group. Third is valine, which carries CH3 group as the substituent. Next is leucine, which carries CH2, matlab homologous, CH2, CH3, CH3. Next one is isoleucine, isoposition, as you all know, it is CH, CH3, CH2, CH2. So in this way, we classify the aliphatic type of non-polar amino acids. Next term is the classification of amino acids that are non-polar and aromatic, means they do not carry any charge but they carry benzene ring in their structure. So they are three: phenylalanine, tyrosine and tryptophan. Phenyl means benzene ring, alanine means alanine wala amino acid. So you have to introduce alanine amino acid over the phenyl group. Phenyl means benzene ring. So here we reverse the chain. Similarly, CH NH3 positive CO negative, and the alanine carries the CH3 group. Here H is replaced by phenyl ring, so it is CH2 and phenyl ring. In a tyrosine, similar to phenylalanine, just it is a OH group containing, so we apply OH group below the chain. Next one are the tryptophan. It is quite complex. First what you have to do, you have to draw the general structure, that is NH3 positive, C, H and CO negative. This is the general structure. Then you have to CH2 as its substituent. Then you have to make two rings. First is hexagonal, second is pentagonal. Hexagonal, which has 6 carbons, pentagonal, which has 5 carbons. Means one ring is of benzene and other is a 5-membered ring. Then you have to add double bond and NH group. This is amino type of a chain. This is quite specific and special kind of the chain that we observe in only in the aromatic type of amino acids. So in this way, we explain all these types of the amino acid based on the polarity of the chain. Now it's time for second type of classification based on the their standard structure. Okay. So on the utility or the requirement, amino acid can be of two types. They can be essential or can be non-essential. The amino acids are said to be essential only if they are not synthesized in our body, or we should take these amino acids in our diet. So essential amino acids are those which our body cannot synthesize or should be taken in our diet. So they are part of a human diet. They are said to be the essential type of amino acid. Other which our body can synthesize and we do not have any need to take them in diet, they are said to be the non-essential type of amino acids. So essential one are those which are necessary to be taken by in our diet, and the essential one, non-essential one are those which are not necessary to be included or to be part of our diet. Okay, so we have done two classification based on the side chain, based on the polarity. Okay, that is in the one. Third classification, it is based on the diet requirement, that we need these amino acid in our diet or not. Okay, so now we are on the third classification that is based on the type of the existence, that is of amino acid. They can be standard or can be non-standard. As you know, we have usually more than 30-100 different amino acids in nature. Out of those, we have only 20 amino acids which are commonly found in our protein, and those standard alpha amino acids are said to be standard alpha amino acids. And all other amino acids which are not found in our protein directly, but they are actually non-protein type of amino acid, they are said to be the non-essential amino acid or non-standard amino acids. So standard amino acids are 20 amino acids which are encoded by universal genetic code. Other are non-standard which are not incorporated inside our protein. So non-standard amino acids are those that are Celinocysteine and Pyrolysine. Standard amino acids are 20 which carry the alpha chain and they have the alpha carbon in their chain. The general structure, as I explained you in the previous lecture, it is same to that. Now it's time for the non-standard amino acids. So in nature, the 21st amino acid that is said to be non-standard is a Celinocysteine, because it is just similar to the cysteine, but the sulfur group of cysteine here is replaced by selenium element. So it is similar of cysteine, but it is here we replace the sulfur of the cysteine by the selenium element. Okay. Next, the Celinocysteine is not the product of, it means we cannot synthesize the Celinocysteine by the general process of translation, but it is directly inserted into any kind of polypeptide chain. Means it doesn't carry any genetic code as the other all amino acid carry. They cannot be coded by the genetic code, but they are directly involved or incorporated during the translation. Okay, so next is Celinocysteine. Celinocysteine is charged on a special tRNA molecule, that is a special type of genetic. About this you will study in your 12th class, that आपको तब पता चलेगा कि ट्रांसलेशन होती क्या है. अभी के लिए सिर्फ इतना है कि ट्रांसलेशन कोई ऐसा प्रोसेस है जिसमें हम क्या करते हैं, प्रोटीन सिंथेसिस करते हैं या अमाइनो एसिड बनाते हैं, किससे, आरएनए के मॉलिक्यूल से. ठीक है, उस मॉलिक्यूल के ऊपर क्या होता है, कोई ना कोई कोड होता है न्यूक्लियोटाइड्स का. अगर वो कोड अंडरस्टैंड करता है कौन, आपका टीआरएनए, तो उससे क्या बन जाता है, आपका अमाइनो एसिड बन जाता है, वरना नहीं बन जाता. उसके लिए एक स्पेसिफिक कोड होते हैं ATCGUG, मतलब इट इज़ वेरिएबल. तो यहाँ पे इन्होंने सेलिनोसिस्टीन के लिए जो कोड बताया है वो कौन सा है, UGA. वैसे तो आपको पता है कि UGA एक स्टॉप कोडॉन होता है, पर सेलिनोसिस्टीन ऐसे ही क्लासीफाई करता है. ओके, सो नेक्स्ट क्लासिफिकेशन हमारी अमाइनो एसिड्स की है भी, या तो वो क्या होंगे नॉन प्रोटीन, या फिर क्या होंगे मॉडिफाइड, या फिर क्या होंगे नॉन अमाइनो एसिड. मतलब कुछ अमाइनो एसिड ऐसे हैं जो क्या है, अमाइनो एसिड है ही नहीं है, वो क्या है, नॉन अमाइनो एसिड है. कुछ ऐसे हैं जो प्रोटीन का पार्ट नहीं होते, और कुछ ऐसे हैं जिन्होंने अपनी प्रॉपर्टीज़ क्या कर ली हैं, चेंज कर ली हैं. सो पहले कौन से हैं, नॉन प्रोटीन. ऐसे अमाइनो एसिड जिसको हम कोड किससे करते हैं, जेनेटिक स्टेट में, जेनेटिक कोड से, जैसे कि सिट्रोलिन हो गया और निथिन हो गया, पर वो किसी प्रोटीन का पार्ट नहीं होते. सेकंड क्या आगे, नॉन अमाइनो एसिड, मतलब वो अमाइनो एसिड ही नहीं होते, अमाइनो एसिड के तौर पे काम ही नहीं करते, तो वो क्या होते हैं, कोई ना कोई हार्मोन के तौर पे काम करते हैं, जैसे कि गाबा, मतलब गामा अमाइनो ब्यूटेरिक एसिड, ये एक क्या है, न्यूरोट्रांसमीटर है, और क्या है, बीटा अलेनिन. उसके बाद आगे मॉडिफाइड, जो अपने आप को किसी भी तरह की एडिशन या डिलीशन की वजह से क्या कर लेते हैं, चेंज कर लेते हैं. तो वो कौन से हैं, हाइड्रोक्सीप्रोलिन और हाइड्रोक्सीलाइसिन, जिनमें हम क्या करते हैं, हाइड्रोक्सिलेशन, मतलब OH ग्रुप को क्या करते हैं, ऐड करते हैं. ओके, सो द लास्ट क्लासिफिकेशन वो किसके बेसिस पे है, आपकी मेटाबॉलिक क्लासिफिकेशन है. मतलब आपकी जो मेटाबॉलिक रिएक्शन है उसके बेसिस पे हम क्या करते हैं, अमाइनो एसिड को क्लासीफाई करते हैं. ओके, सो उसमें हम क्या करते हैं, आपके अमाइनो एसिड कैसे कैसे होते हैं आपके, एक तो ग्लूटोजेनिक, जो क्या बनाएंगे, आपकी ग्लूकोज फॉर्मिंग बॉडीज बनाएंगे. ठीक है, और एक आपके कौन से होते हैं कीटोजेनिक, जो आपकी क्या बनाएंगे कीटोन बॉडीज, और एक क्या होते हैं मिक्स्ड, जो दोनों ही टाइप के क्या बनाएंगे प्रोडक्ट बनाएंगे। तो इन दिस वे आपने इस टॉपिक में ज़्यादा ज़रूरी क्या करना है, एक तो क्लासिफिकेशन करनी है कौन से वाली, कौन से पोलर हैं कौन से नॉन पोलर हैं, कौन से चार्ज्ड हैं कौन से अनचार्ज्ड हैं, बस। और फिर एसेंशियल और नॉन एसेंशियल को क्या करना है, याद करना है भी, मतलब कौन कौन से हैं। दैट्स ऑल फॉर टुडे, ये लेक्चर कंप्लीट हुआ। नेक्स्ट टॉपिक में हम क्या करेंगे, प्रॉपर्टीज़ और सम ऑफ द फंक्शंस ऑफ द प्रोटीन्स। थैंक यू। और आप प्रिपेयर करते रहना, जो चेन्स हैं उनको ड्रॉ करते रहना।